Well, we at least have an image.
Sorry?
I said at least there's an image.
Yeah. How did you color the samples here?
How do I color them?
Huh?
I use a program called Adobe Lightroom.
Oh.
I don't know where the mouse went. Oh, maybe it went over there. Okay, so now we're live. So today we're going to be looking at a sample from, well, several samples from Lake Tanganyika in Africa. And I'm primarily looking for some little gomphonemas of high quality that I can take a picture of for Tuma, who's a PhD student at the University of Arizona who's working on these samples.
Cockroach.
Yep, it's Cockneys. And all these little fragments that are here that are laying around on the screen are pieces of Nitsia, which is pretty
Sure?
much, yeah. It's Nitsia, they're all little pieces of Nitsia,
Oh.
which is pretty normal for Lake Tanganyika. And then these little perfect spheres that you're seeing are microspheres. They're artificial pieces of, I don't know, I guess they're plastic that we put in the sample so that we can identify concentration differences from one sample to the next. And I do have help today. You want to be on the camera or are you hiding from it?
No.
No, you're hiding or no, you're not?
I'm not hiding.
Oh, okay. It's slightly out of the field of view. There. Sukanya's with me today. So she's a PhD student in my lab. And hello Strack, and hello Alex Baldwell, and hello Luan. How are you doing? And also, of course, hello Pacific Plankton. So Sukanya's hanging around on the outside here. I guess she's trying to help me identify genera.
Yeah.
Yeah. In case I need help.
I know you don't need any help.
I don't?
Yeah, you are working on this data.
What's this one?
This most probably in cyanine
You don't know?
or navigator. It's broken one.
It is broken, but it has a slight S-shaped curve to it.
Yeah.
So it's gyrosigma.
I don't know that one.
Yeah. That's probably why you didn't get it. You don't know that one yet.
Yeah.
Yeah. See? Nitsia?
Oh.
They're just everywhere in the sample. That's what Tanganyika's like. Almost the whole sample Nitsia, and then a few little things occasionally. Or every once in a while, there'll be samples that are just filled with giant cyclostephanos or something like that. Stephanodiscus sometimes. It's a pretty depressing sample if you wanted to not have to count Nitsia, which is usually the case for me. Usually when I see Nitsia, I'm just like, someone else can count that one. No thanks. It's not actually a very good sample either. It's like clumpy.
Mostly are broken here.
Yeah, all the Nitsia are all broken. So if you were counting this sample, you would only count the
Broken fragments.
the ones that have valve ends,
Okay.
and then you'd have to cut them in half. So now you'd have to count twice as many Nitsia as you were counting before,
Oh.
right? To get the same number out, because you're counting half at a time or something like that.
Mm-hmm.
Oh, there's like one little diatom out here by itself.
Yes.
And it's a Nitsia. Of course.
Completely different from light microscope and through an SEM, the Nistria.
Well, it's the same organism. They just look different, right?
Yeah.
So, but I would say that's true for almost all the organisms. They look pretty different in SEM.
Yeah.
Nitsia are slightly less hideous in the SEM, but only slightly.
That's why you didn't like Nistria.
It's a Gomphanema, but it's a broken one. We're looking for that thing, only I want the whole valves.
How did you identify broken fragments?
How do I identify them?
Uh-huh. Is it Storcella?
This is a Nitsia.
Oh. Now I
Nitsia.
hate Nistria.
Well, I don't know what to tell you about that. Join the team.
Yeah.
Nobody likes Nitsia. Actually, I do have some colleagues who like Nitsia.
Like in light microscope, it's easy to identify.
No. It's easier to identify in the SEM, in my opinion. Nitsia?
It's not Nistria.
This one?
No.
No, this one's a Fragillaria, some type like
Storocera.
Yeah, or Starocirella.
And at least one.
Can't tell for sure. See? Nitsia, Nitsia,
Oh,
Nitsia.
yeah, now I understand.
That's I think that's a Starocirella. It's not a very good sample in that it's got clumps of stuff together, and then fragments everywhere, right?
Mm-hmm.
So we're not going to win any awards for prettiest photographs out of this, probably. I don't know that I normally would. That's a Nitsia. Again, little Nitsia. There's sort of long ones. There's asymmetric ones in here. And broken ones.
Mm.
And then a few of these little guys. But I think most of the samples broken fragments of Nitsia.
Okay.
As to how I identify them, I don't know. I
Yeah.
just do. I'm used to seeing them, so that's a company.
Gomphanema.
Yeah. but it's broken.
Yeah.
And also a little dissolved up here at the top. Let's see. I missed comments because I'm looking in my samples. Greater Jason says, I saw you on Pacific Plankton and I'm very excited for this stream. Oh, okay. Welcome in, Critter Jason. This is my scanning electron microscope lab. And yeah, I'm doing serious science today. Well, plus or minus. And then, yeah, there's an asymmetric Knitsia here in the sample. And Pronature, hello. We're having contests to see who can make the most angelic images. Really, it's more like an Easter egg hunt than anything else right now, because I'm trying to find something that's, well, somewhat common in the samples, but not extremely. Also, I was trying to tweak the settings to make the image quality a little bit better, and I think it made it worse, because the auto programming on here was having a lot of trouble this morning trying to, well, do anything. So
It's all of the same.
this? It's a Knitsia.
Oh, gosh.
I think there might be some Alicosira hidden in here, but that's a Knitsia. I'm going to see if I can fix some of the image quality, because I can't bear it the way it is. So this is our problem, is it wobbles really off. You can see how much it's moving up and down, right?
Mm-hmm.
Now a bit less. It's worse.
So that broken fragments of Nishtia, what they infer about the lake?
What do they tell us?
Mm-hmm.
Well, in Lake Tanganyika, the Knitsia are mostly planktonic, especially these long skinny ones. And they represent periods of, where we see them in the sediment record, they represent periods of upwelling. In this case, they probably are telling us something about nutrient upwelling. And the reason that they tell us something about nutrient upwelling is normally they wouldn't be preserved because they're very lightly silicified. And so in order for us to find them in the bottom of Lake Tanganyika, it usually means that we had to have a lot of them so they could overwhelm the ability for the water to dissolve them. So think of it like really high concentrations of Knitsias. They're all being dissolved as they're falling through the water column. But if we have a whole bunch, a lot of, like a high volume of them, it can't dissolve all of them before they hit the bottom, right? So if we had a fairly limited amount of them,
Then it get dissolved.
they would all completely dissolve, right? So the fact that they're broken could actually indicate a couple of things. One of those is that there is an organism in the lake, which I think it's a copepod, that eats diatoms, and they can actually have the ability to sort of chew it up and break them up. And so I think they actually, like as they chomp on it, destroy some of the Knitsia. And so what we see on the slides are these samples are probably clumps of material that came down in the form of little fecal pellets, like they rain down from above. And
This is Nishya.
this is Knitsia.
Oh,
Yeah. At this point, it's a good guess. You could just go Knitsia, and you'll probably be right, like 90% of the time. So the fact that they get eaten probably results in some of them being broken. So we're seeing little fragments of them. And the fact there's another Knitsia.
Yeah.
The fact that we are finding them at all in the bottom might be a result of the fact that those fecal pellets actually fall through the water column a lot faster because they have more mass. And so something like this, where we see them all clumped together, so this is like a whole big mass of broken Knitsia, is probably just a little bit of poop.
Oh.
Probably some sort of copepod poop. So now it's a poop stream.
Oh.
I know Pacific Plankton was waiting for me to have a poop stream, so. So all these little dots you're seeing are microspheres. And normally we put them in the sample so at a specific concentration, so that when we see a lot of them, we know that the diatoms that are present are low in abundance. And if we see very few of them, it means that relative to the number of diatoms, that the concentration in the sample would be higher. Concentration of diatoms, I should say. It's the same concentration, at least in theory, of microspheres in every sample.
Okay, so depending on microspheres, we can understand the relative abundance, means the in general relative abundance of the diatoms.
Because if we have the same number of diatoms in every sample, and I add a fixed number, like 100 microspheres, then every sample we should have the same number of diatoms to microspheres.
Oh.
But if I add the same 100 microspheres, and now I have twice as many diatoms in each sample, right? Then the ratio of microspheres to diatoms would be 1/2.
Yeah.
Right? And if I were the opposite, if I were to actually have more microspheres than diatoms, it tells me that the concentration of diatoms in that sample is lower. Right?
Yeah.
So we can actually use the microspheres as a way for us to get a sense of changes in concentration. And the way that that's useful for us is, as long as we put the same amount of sample in every one, like each sample we put in the same number of microspheres, and we weigh the samples so that they have the same mass, we can get an idea or an estimate of changes in abundance of diatoms.
That's why you told us to count the microspheres.
That's why we count microspheres. Yeah, so the real value is that if from one sample to the next the lake becomes more productive, then there'll be more diatoms and fewer microspheres. And if it were to become, for some reason, more dissolved or less productive, the opposite would happen.
It's inverse relationship they have.
Yeah, so it's a way for us to basically just sort of get a sense of changes in productivity. Diatoms are primary producers, so they usually don't do better in the system unless there's more nutrients in that system. So I think I'm going to take a picture so that I can interact a little bit with chat.
It's Gumphney.
It is Gumphanema. So this is the one that we're trying to get the images of. It's Gumphanema clevi. And In these samples, it's dominant. In the microscope counts, which is kind of interesting because if we look at the samples here, mostly what we see are Knitsia, but almost all the Knitsia are just fragments.
Yeah.
So if you were counting them, you wouldn't count very many.
Mm-hmm.
And the reason that we don't want to do like count each fragment as a whole diatom is one Knitsia could be fragmented into, 40 tiny pieces. We don't know how many, right?
Oh.
So in that sense, we would always want to count either only whole valves or only valves where we have the valve end, because there's only one of those,
So
or two of them actually. But
if any centric diatoms, if we have only the center part but not the edge part, then we should count that one.
yeah, yep. Center parts count as a whole diatom because there's only one of those. I wanted to unclick from that thing. Okay, so let's see. Pacific Plankton's been handling the questions in chat. It's the lady with the amazing voice, yeah. Pacific Plankton is here. So in my streams, Pacific Plankton is my moderator, and in her streams, I moderate.
Oh.
So, what happens when you change beam intensity? So beam intensity determines how much of it's basically the spot size, or how much of the sample is being hit by electrons. So if we made it a very narrow beam, then the resolution of our image would increase, but the amount of information coming from the sample would decrease, because it's sampling using a smaller, like a finer point beam. And as a result, we'd have to slow the beam down to see the image more clearly. So usually there's some sort of a trade-off between speed and beam intensity. Sorry to ask such a rudimentary question, but nanometers, oh yeah, it's 1,000. Oh well, yeah, 1/1000th of a meter, or 1,000th of a millimeter. Yeah. Yeah, you're not actually disturbing me. Go ahead and ask questions, and just whenever I get down to the point where we can take some pictures, then I'll answer them. So for no more Grim, everything you're seeing is live. Where I'm here, the image is there. I'm talking about it as I manipulate it, so nothing is recorded except for when we're done. It's being scanned live. Yeah. So it's stable, but relatively big images. Yeah, it's drawing images at 1280 by 1280, and so it just takes a while for it to scan it sometimes. All right, I need to change folders. 2A. And there's still a bunch of questions I haven't answered. I saw them, I just will get back to you. Thought I saw another Gonfanema over here. Yeah, it's a pretty poor sample, but there is one. There's an external view of the same diatom we were just looking at, Gonfanema clevi. And somebody asked who's the person I'm talking to. So in the room with me, let me see, I can fix this for a second so you can actually see her.
Hi.
Sukanya. She's a PhD student who's working in my lab, and she's still learning diatoms, but getting the hang of it, I should say. Right?
Yeah.
So, this is not Sukanya's project at all. So these aren't samples or diatoms that she's ever seen before. So she's just sort of hanging out with me this afternoon. And but it's useful because I think she hasn't seen a lot of diatoms in the scanning electron microscope before. And it's a nice way to sort of get some training, right?
Yeah.
So
I mainly observe your talking so that I can talk in the same way any speaker.
oh, you're also working on your English? Is that what you're trying to say?
No.
By listening to my English?
No. Like in how you talk for one thing for a long time.
Oh, my method of presentation?
Yeah.
Well, that's good because you probably shouldn't be using me as a model for English speaking.
I can use. You're better than me.
Marginally.
None of our better.
My Hindi is pretty bad though. I don't speak it at all.
You know Hindi?
No. No, not at all.
Okay. You know Bengali?
I don't know any Indian languages actually.
Okay.
Sorry.
Thank you. Sometimes I speak Bengali.
If I started speaking in Hindi, you'd probably freak out, right? Be like, wait a second.
No, I will enjoy then.
You would?
Yeah. Do you speak Spanish too?
No.
Okay.
Why do you speak Spanish?
Yeah, sometimes.
I learned French in high school. And,
So you know French?
well, I learned it in high school. So it was a long time ago. And I don't use it very much. But I can probably still read it. Or read most of it. But I can't really speak it. I can't conjugate verbs anymore or anything like that. But I know, still remember most of the words. So,
That's good.
but I would speak like, very poorly.
Like
So-
my Spanish, I also speak very poorly.
Well, I'll talk to you in French, and then you can talk to me in Spanish, and then it'll be perfect.
Are these two languages same?
No.
Then how can I understand? Maybe you were asking about my name and I'm telling my home address.
I would probably ask you, where's the library? And let's see, what else do we know? Yeah, what's your name, I guess. I know that one. The funny expressions that we learn when we're learning languages that don't help you later.
Yeah.
Like you need to know where the library is for some reason.
Like I don't have any culture right now with Spanish, so most of the words and the structure, how to form a sentence, I forget.
Well, you could just talk with Laura.
Yeah, I tried sometimes.
Does she make fun of you about it?
No.
No, exactly, because she wouldn't. This is that same Gomphanema, but it's in girdle view. You
And
see
it's
everything,
broken.
a little bit. Everything in the samples is sort of covered with a little bit of mud too. So the images are kind of like, I don't know, not as interesting. In part because they're not as clean. And then of course like I said, we're seeing a lot of just sort of chunks and pieces of things, which doesn't help.
Yeah.
So,
So it would be difficult for counting because
well, the good news is Tuma has already counted these samples. So nobody has to count them anymore. Tuma is, as I mentioned, he's a PhD student working with Andy Cohen at the University of Arizona. And I was on sabbatical at the University of Arizona 2 years ago. So my family and I all went to Tucson, and we stayed there for the better part of 4 months or so. And I worked during that time to train Tuma how to identify diatoms from these samples. So we actually had these samples there. And then basically I would sort of explain to him what he needed to know to recognize the species that were in his samples. And then we also had some guidebooks from Tanganyika, which were helpful to build on. But so he's already managed to do all the counting for these, and we've got a publication that is that Tuma wrote. So we had one that was published last year on this material from Tanganyika that Mike McLew wrote, and that I co-authored with him and Sarah Ivory and other people. That was in Science Advances. And then Tuma's article includes information from this core, which is Tanganyika 2A, which is the one that we actually presented in the Science Advances article, and then another core, which is 6A, and compares them together. So this is a pretty interesting article, and then that we wrote, and then Tuma sort of builds on it and talks a little bit more about the structure of the lake that we're reconstructing, and some of these sort of interesting intervals like these that have a dominance of other things besides just Nitsia. So I'm going to jump over to sample 1 and see if it is any better with respect to finding Gomphanema that are not just fragments and not covered with junk. So I don't know that we're going to find a lot of really great samples based on what I've seen. A lot of what I'm seeing in these are just like little bits of debris and then clumps. So less than ideal for SEM work. I did a little bit of preparation. I rinsed them, but I probably should have tried to sonicate these samples before putting them on the SEM, see if it could break some of these little clusters of stuff apart.
Yesterday you collect some water for diatoms, I think.
Yeah, yesterday Laura and I went to a little koi pond on campus.
Where is it?
Where is it? Out in the middle of the quad. I don't know how to explain to you where it is. I mean,
Next time I'll go.
Yeah, you can come along. We were really just sort of exploring stuff to see what was in it, and I needed to have some material to look at for my evening microscope stream. And then it turned out that we didn't even, I mean, there was stuff in it, but it wasn't that exciting, so I ended up using some other material.
Oh.
It's another diatom. This one is a placanese.
Yeah.
And I'm going to work on making it less blurry for us. 1 second. There we go. It's a nice clean image of it. And going to set it. So you can see what happens here is I change the speed of the SEM, and as you change the speed, the resolution of the image changes. So now it's scanning slower, so we get a nice high resolution image, but it takes longer for it to actually draw the image for us. So and the one thing that the SEM is pretty good at is adjusting for the brightness and contrast. So I usually just let it do the auto brightness and contrast. And this image is probably okay before I started telling it to check it, so it probably won't change very much. But there's kind of a bright boundary around the diatom that you can see it's making a little bit darker. And that's bright because it's pointed upward at us. So we're looking into the inside of one half of a diatom cell. And the bright components are bright because they're more of the electrons from that surface are being captured by our detector because they're pointed up at us. So, okay. So I can take a picture and then I can hop over to chat and see what's been going on because I feel like I'm not getting very much time with discussing things. Okay, so Pacific Plankton wants to know how do you say diatom in Hindi?
It's diatom.
It's just diatom.
Mostly scientific name doesn't have any specific name in Hindi.
No More Grim wants to know, do I have an age for the sample? Yeah. Do I know what it is off the top of my head? No. But we do have ages for this interval. I think it's several hundred years old. It's not super ancient. The 2A core goes back about, I want to say 2,000 years or something like that. So, hello, Devil and Mrs. J, and thank you for giving her a shout out. You got the rest of your diatom sticker order yesterday. Great. Let's see. There's a little Redbubble site for anybody who's interested in getting yourselves some diatom stickers and or other things. And let's see. So those are, in some cases, the stickers are all my drawings, where I've taken SEM images and then rendered them into a graphic. But then there are also some SEM style images that you could get as whatever on there. Yeah. And you got a new laptop from your dad on which you put the stickers. Excellent. It's a good place for them.
Yeah.
Freshly vandalized by science. So this isn't a Gomphanema, it's a Placanese. But it was pretty, so I thought we should stop and take a look at it. Plus it gives you a good sense of all of the Nitsia fragments that you can see clumped around on the entire margin of this thing. So the sediment is almost entirely dominated by little chunks and pieces of diatoms. And we can go back to a live view. You can see here's a whole big cluster of them. And I think that's actually a Gomphanema right there. The one that we're looking for, just broken a little bit.
Inside, nearby the Gomphanema, that long one.
This thing?
Yeah.
Sponge beetle. Yeah. Ah, all right. I see what you're doing. Trying to time it so it sounded like you were coming out of my mouth. Yes, this is sponge spicule. There are a bunch of sponges in the Tanganyika material, and so we'll have plenty of opportunity to check some of those out. There's a little cockneese in here, and I think that's a little calanice. Yeah, it's a calanice. Just a fragment of one.
Oh.
And over here in the corner, that's a
Oh, look.
Cyclostephanos.
Oh.
Yeah. Here's another big sponge spicule right here.
Yeah.
So that one is bigger than 100 microns, which means it's a megasclere,
Yes.
which we learned earlier this semester in Dr. Yost's class,
Yes.
of which I'm a part.
It's gone.
That is a Gomphanema.
Good news, everyone.
Hey, we got a new follow. I should go look at that. It's Holtzy. Hey, Holtzy, thank you. Thank you for the follow. Now you'll be able to tell when the scanning electron microscope is being used for streaming. So it's great. You can check in with us every time. Or anytime. Another Gomphanema, also fragmented. A little cleaner than some of the other ones, though. At least the image is nice and clean. The diatom doesn't have a pile of junk on it, which is a positive sign.
Yeah.
And we got another follow.
Yeah.
Oh, it's No More Grim. Ah, thank you for that, No More Grim. I think because it's broken, I don't want to image it, but because it's so clean, I do want to image it, so I'm torn. I'm going to rotate it. So one of the things that's nice about the scanning electron microscope is, like, down below our image, you can see into the chamber itself. And when I rotate it, you'll actually see the SEM recalculate its position and rotate it around. And I think it's probably like a 330, maybe? We'll change the rotation, and then you'll see it's recalculating the position. And the reason it has to... That was a pretty good guess on my part. I think I went a little over. The reason it has to recalculate is because our sample is on the outside edge of this carousel. And so if it turns the stage, it just turns the middle of the carousel. And then that would mean our sample would just then disappear from our field of view. So it has to figure out and project where our sample would be, and then it will turn the stage and then move the stage to that position so that we don't actually lose the sample. And that is actually so this is the raphy.
Raphy, but that dot.
The dot is a stigma.
Okay.
On a Gomphanema, it's a stigma.
Okay.
Yep. So that's just a little hole that passes through the cell wall. And then if we look up at the top, so I'm just going to ink it down a little bit. Up here, that's the apical pore field. So that is how this diatom attaches to a substrate. And then if it crawls around on the substrate, it'll use the raphy. But if it wants to be like attached, it attaches through a stalk, and it puts the stalk out through this apical pore field.
That's what we read from that paper, high profile and low profile.
For high profile diatoms, usually they have a stalk, and then they're out floating on the stalk, so they're up above the surface, quite high usually. Well, relative to the diatom. The stalk might be half the length of the valve or longer in some cases. What? It's dangling? Hey, dangling, how you doing? There's a voucher for free stickers on Discord if anybody was wanting to add some to future orders. Yeah, I saw that. Thank you for posting that, Devlin, Mrs. J. And it's nice to see you, Dangling. You know what? We were puzzling together last night, Dangling, and you missed it. So. Who's talking is Sukanya? Who's a PhD student in my lab. One of, I don't know how many people in my lab now. 8? I think there's 8 people in my lab. Yeah. So. Yep. Closed captioning says ethical Ford field. Yeah, sorry, that's probably the mask.
Yeah.
Apical. Oops. Ford field. Ethical Ford field is great, though. I like that. Yeah. Oh, I guess Pacific got to it. If two diatoms look the same, how can you tell they are the same species, or you just compare the way they look? Or is there any further research? So, if two diatoms look the same,
then we need to count the striae of that diatom.
so yeah, you can try to compare them by looking at the density of the striae, so these are striae. This is a row of areolae called striae. And you could look at the density of the striae, like so for every 10 microns, how many striae do they have? You could look for the position of key features, like the terminal raphe ends and the proximal raphe ends and the stigma. You can count the density of the areolae.
Yeah.
You can measure the length and width, and also you can do a bunch of shape analysis. So if they're kind of close, you could use all of those tools. And then if you have a living material, which these are all fossil, not that old, but they are fossil. If you had living material, you could check them through their DNA. So like as long as we know, we have known species that are voucher species for the DNA, then you could compare them directly to those. And one of the problems with that is, of course, the DNA is not going to help you determine whether it's a new species or not. That's something that has to be determined, by decisions that you make. It will tell you how close it is to an existing, like a known species, and if it's identical, you should be able to tell. But a lot of times it's hard to tell, how far two things have to be separated genetically before they actually are different species. So thinking about it like that, right? Especially if they look similar. If they look dissimilar, then, maybe it's a little bit more easy to do. So it's usually good to have both morphological and genetic information to help you figure out whether you think something should be classified as a new species. And then, so because I mostly look at fossil stuff, we primarily just use the morphological information, because we don't really have, typically, the DNA that goes directly with whatever organism. It's not going to be emplaced in the cell. In this case, we don't even have whole cells most of the time, so the idea that we would actually have a whole cell with the DNA within it is very unlikely. So here's a big sponge spicule, for example, and you can see also here's a bunch of Nitzia in a clump, right? And see how bright that is up there?
Yeah.
It's because it's a high point, and then the electrons are basically, more of the electrons are reaching the sensor from that high point. So it actually would then distort the rest of your image a little bit to try to take a picture near it, which is why I'm just moving away from it. It's like a little bright hot spot, and it's
Something is there.
this little guy.
No, that from the clump.
Oh,
Yeah,
here?
Yeah.
Yeah, there is a larger piece of something, a fragment.
Oh, it's.
Hey.
Shit.
This?
No.
That's a raphe.
Yeah, that's a raphe,
But the raphe's running down the middle of the valve, so it can't be Nitzia.
Oh, yeah, right.
So it's probably a Gomphanema. I don't know. I can't tell, actually, from that bit, but I think it's probably Gomphanema. A bigger one. Not the clevi, something else. Do you see this, like, giant clump of stuff, right?
Yes.
Just didn't get separated very
There
well.
is one ensign.
Yep, that is an ensynema in here.
Yeah.
And also a phytolith,
Yes. By bilobes.
a bilobate,
Yeah. Bilobate.
and I believe that is a gumphonema right there. Just got some junk
Yes.
on it. That's a gumphonema. You can see there's Knitsia all over everything,
Yeah.
though. Clumps of Knitsia everywhere. Typical Tanganyika sample.
So all of this Tanganyika sample having this kind of lumps.
No, I think that's just more. This sample in particular has a lot of clumpy stuff. I just meant it's all full of Knitsia. That happens in Tanganyika a lot. So usually if we have something else like Alicocera or Cyclostephanos, it's because all the Knitsia have dissolved, if there's a lot of them. So here's a internal view of a Cyclostephanos
Yeah.
in Lake Tanganyika. The Cyclostephanos is, they call it CF Damasii. It's the closest thing. I don't know why somebody hasn't put a name on it if that's the case, but this is actually really useful if you're trying to figure out what Cyclostephanos looks like. So what happens with Cyclostephanos is the interfascicles, which are these sort of solid pieces or costi-like structures that go between the striae, they come down to the manto photoportula, which is this thing, and then they bifurcate, and they continue all the way out to the valve margin.
Okay.
So that separates it from Stephanodiscus. And Stephanodiscus, typically, the costi stop at the mantle photoportula. So that little hole would be where everything would stop, and then there wouldn't be any extra costi stretching beyond it. It seems like a really tiny difference, but it makes a big difference because it actually makes these into sort of chamber-like structures.
But that difference only can be identified through SEM.
Pretty much, yeah. So you can see some of it in the light microscope, but it's pretty uncommon to be able to see it clearly.
Is it a Alicocera?
These?
No.
Knitsia.
Oh.
Just a piece of a Knitsia.
Okay.
I haven't actually seen any Alicocera. Maybe I saw a fragment of one earlier, but I haven't seen any whole Alicocera. I did see this giant guy.
Yeah.
There's an external view of that same Cyclostephanos. They're huge. Normally in these samples they can be, this one's about 40 microns across, but they can be like 100 microns.
Oh.
They're really quite large. I think some of them are larger than that. Had some samples from Tanganyika that are just filled with them as well. That might be an Alicocera. Yeah. There's an Alicocera. There's a little fragment of gumphonema. Yeah, everywhere. All in clumps.
Yes.
Some of these samples, I think when Tuma first counted them, there wasn't enough like non-clumped diatoms for him to actually analyze. So I think we had to remake some of them, or resend them to him, and he remade them because they were all just clumps like this. Sponge. Yep, a bunch of them. Here's one, here's one, here's one. It's like for a minute I thought this sample was going to be a little bit better, but it's just all full of clumps.
Mm.
So still not great. But probably one of those pictures that I collected is good enough for Tuma. Yeah, for the company. We weren't trying to identify anything. In fact, I'm just mostly trying to get some pictures for Tuma for his publication. So he has a Diatom of the Month article that he's been composing. And I think he just wanted to have something to go along with the article. So it helps if it's pretty, but I guess if we don't have anything glorious, that's fine. Not every SEM image is going to turn out to be glorious, unfortunately. I can check this. I think this one is still Tanganyika. And then I have some more samples in here from Addie's stuff from Clear Lake.
Wow. So it's Addie stuff now?
No, this is still Tanganyika.
Oh.
That's a Cymbella. Cymboplura,
Oh, okay.
I think. Cucumis.
I didn't see it in my sample.
No, probably wouldn't. And then you can see here's a big Ensynema, and here's a little, I think that's a stuff. Stephanodiscus or Cyclostephanos, maybe. It does go, it looks like they go all the way to edge, so it's probably Cyclostephanos. Maybe a different species, though. Some other big diatom here. There's a Cerarella.
Yes. 2 bulbs are present. Both of bulbs are present?
Actually, I think they are both present, yes.
Yeah.
I can't tell what's going on the other side, but... Sponge spicule.
Mm-hmm.
This sample has a lot of benthic organisms. This is Capartogramma, which is a genus that's mostly in Africa and not very well reported anywhere else, although there are some instances of it in North America.
Tell the name in it?
Capartogramma?
It's present in one of my samples.
Yeah.
But only one.
Yeah, they're pretty uncommon, but they are found in marine samples or like rivers near marine settings, so salty rivers.
Yeah.
Yeah. So probably would be something I would expect to find in your samples. But prior to about, I don't know, maybe 15 years ago, people hadn't even found it in anywhere else.
That one?
They'd only found it in Africa. They thought it was
Oh.
only African.
Okay.
I don't think it was very widely reported as being outside of Africa, but I think it's mostly because people just hadn't seen it because it's rare. Well, I think that's all Tuma's gonna get, based on what I'm seeing here. So let's jump over and take a look at some of Addie's samples.
Yes.
They've got a lot more diatoms in them. And you want to look at this one?
Yeah.
Is that where you're pointing at? Telling me what to do? It's a...
Name.
Just say it. You're not going to say it?
Scared about now.
Well, you had it right.
Can I see the whole one again? It's navicular.
Yeah, it's navicular.
Okay, something I learned.
The striae have these little lines in them.
Yes.
So, as long as the shape is boat-shaped, it's probably going to be a navicular.
So all navicular species is having the same boat-shaped striae. Here, like.
Lineolate striae, yeah.
Oh.
That's for navicula in the strictest sense. Yeah, they will.
Okay.
They should have lineolate striae like these. It's gonna look like a pretty good picture if I slow it down.
Mm-hmm.
And I can stop neglecting chat for a little bit while that
Yes.
processes. Bluesy, hello! And Little Chook! Hi, Little Chook! It's nice to see you, and you got your new emotes going. Very good. Oh, look, it's Anna. I missed a bunch. What did I miss?
Yeah, from apical portrait.
If two diatoms look different, they belong to different genera. Yeah. Let's see. Mallory is currently teaching my daughter how to swim. So that's why she's not here. But also, she doesn't hang out on Saturday streams. I think she's decided she'd rather sleep. Or apparently teach somebody how to swim. One more question, if it's all right. Are there diatoms which you can't look at with the SEM because electrons could damage the sample? Probably not. The SEM will do a little bit of damage to the diatoms, but it's just at the electron scale, so it's probably way too small to actually, at least at the intensities that we run the SEM at, to cause any damage to the actual organism, like the skeleton. There are some diatoms you can't process, though, so you'd have to just put it in raw. So they're so lightly silicified that basically they fall apart if you try to do anything with them. So, yeah, the closed captions are probably pretty funny. They don't know how to handle science terms. So, and my second vaccination is April 1st, so I'm looking forward to that, and I'm glad everybody out there is starting to get vaccinated. Cyclist of Thanos. Let's see. If you don't know who any of these people we're shouting out are, you should follow them, especially Little Chook and Devil and Mrs. J. Little Chook is an incredibly good artist.
I knew only Pacific Plantain because in that one interview, I think said that time I saw her.
Mm-hmm.
And she is quite nice.
Yeah, she's super nice. She hangs out with us all the time. She's learning how to play Valheim right now on her new laptop.
Oh.
A Diatom of the Month. Yeah, it's going to be Tuma's Diatom of the Month. So, I'll post the link in our Discord eventually. So, yeah. Oh, we're all the way back. We're up to comparative grammar. Let's see. Headshot Specialist, hello. April 5, excellent. And Devil and Mrs. J also does a bunch of really cool stuff. She's been working on some costume art, I believe. This is not Tanganyika, but Clear Lake. She does a whole bunch of crafting stuff on her stream. So, very cool. Just gonna label this Navicula internal. Just add random things to Addie's folders while she's not looking. I'm sure she won't mind that much. There's another one up here. You just want to look at the big things, don't you?
Yeah.
Well, genus?
Navicula.
That's Navicula. Yeah. This is Navicula oblonga, and the other one we were looking at, I think, is Navicula aurora. But
How do you remember the species name?
how do I remember the species names?
Yeah.
Well, they're like old friends to me. I remember your name.
I'm a human.
Oh, really? Are you sure? What does that got to do with anything?
Like, but it's very difficult to remember each species name. Okay, this is what is it?
What is it? It's Navicula again. See? Lineolate striae.
Yeah.
Now we're looking on the outside of the
But the
diatom.
raphe is kind of hook-shaped.
Yes, it does have hook-shaped raphe on the proximal ends, but that's not a big deal.
It's Navicula.
Yeah, I think it's broken though. Look at it got wrecked up here. We could just be artsy and like show the wrecked diatom.
I think this one.
You want the Oligocera?
Yeah.
There's a big
This
sponge
one's
baby.
bigger. Yes, this one.
This is a little chain of Oligocera.
Yes.
And actually this is kind of cool. The girdle band has come off of this part, but not these parts. So let's see, I need to make that number smaller. And still a little smaller. So I'm rotating it around just so that it's corner to corner, so that I can zoom in a little bit farther. With every little bit you zoom in helps when you're trying to make the image bigger. And you get a really nice clean view of
Girdle.
the spines and how they join. So that they have little forks on the tips of them, like
Yes.
a tuning fork. Some are a little bit more spatula shaped. And then you can see these are still wrapped by the girdle bands. So you can't see the pores very easily in them. The angle could be just a little bit better. I'm not going to be fussy about it though, I'm just going to take a picture. It's a little over bright.
So you remembered all the rotation point also, 160, 150, or like here you put every time changing the rotation value.
All I'm doing is looking at the number that's there. So it was at 180. And if it needs to go this way, it needs to be smaller, and if it needs to go that way, it needs to be larger. So I was just doing some math.
Okay.
I didn't memorize those. I was just trying to figure it out mathematically. So I'm trying to guess, is that about 20 degrees or 30 degrees? It was more like 25, but it's fine. It's close enough. If I tilted a little bit more, I'd be able to zoom in like 2 microns, so I feel like it's not that big of a deal. Let's see. Professor, are there any movie, TV portrayals of diatoms that I enjoy? Hey, that's a good question. See, Holtze says that's exactly what an AI would say. I'm a human. You can't trust anybody. Let's see. I've never seen a diatom portrayed on TV or a movie, like as an actual diatom. But I get what you're asking, which is, do people talk about using diatoms or have diatoms appeared in TV shows? And I'll answer yes. If you saw, so for No More Grimm's question, if you saw, or None More Grimm, um, the TV show Dexter, which was on, I think, Showtime for several years about a serial killer who kills other killers or serial killers in Florida. And I don't know which season it was. I think maybe it was 5. They almost caught him, like, because he would take their bodies and throw them in the ocean, and he tied his boat up to the dock, and the rocks that he collected to put in the bags with the body parts so they would sink, he collected from a specific pier, and they found the rocks inside the bags with the bodies, and they looked at the diatoms on the rocks, and they tracked them back to the pier. So they said, oh, those diatoms only live in this one little pier, and they found the pier that Dexter's boat was launched from, and very nearly caught him using that as a forensic tool, which I thought was pretty cool. You see it in CSI quite a bit, like where they show, well, I don't know if those shows are still on, but they used to have like the forensic people. The people on the forensic TV shows can do every part of the job. Like they can take fingerprints, and they can do diatom analysis, and they can create 3D models of skulls, and they just do every part of the job, which is complete garbage. But the funny part is they'll have something they collected from like a water fountain, like body or evidence or something, and then they'll be like, oh, we found some diatoms on these. And then they'll try to pronounce the diatom names, which just makes me laugh because they never get it right. And also, then when they show the picture, it's like stock footage of marine diatoms. And it's like, we found these in a fountain, and these indicate fresh water. And I'm like, no, they don't. So what they really need for those shows is a consultant. Go ahead and hit me up, CSI. I work pretty cheap, and I can give you the right information, and also I'll tell you how to pronounce the names correctly. I'll even give you some phonetic guides. You can ask Jay Renzella. It's something I do. This is, I don't know, SP. We'll just leave it there. I will be happy to work for CSI, whatever, if you guys need my help with your diatom stuff.
So they got diatoms on the fingerprint? Like on
No,
the
no,
finger?
no. They would take it out of the lungs or off the clothes or off their shoes or whatever. Some sort of evidence that they found. So when I was on the stream, I think Addie and I were streaming on Wednesday together. We kept seeing little green algae bits like these, pediastrum. And that's a pediastrum. And normally they're the whole skeleton of the organism is a colony. Each one of these is this little horn-shaped things is one cell, and usually we don't see them because when we process them, they dissolve or whatever, they're decomposed. Mm-hmm. But for some reason, in Addie's samples, we found a bunch of them. I keep finding them. Here's a giant phytolith. A long cell phytolith.
Crenulate.
Yeah, there you go. Crenulate. See? Sukanya had to pass a quiz. How'd you know the name of this one? How'd you remember its name?
I learned it.
See? That's how I learn all the diatom names. Same way. I saw the Symboplura up here I wanted to check out.
Yes.
Usually these have kind of interesting pores. I don't know if this one will. Sometimes they're really simple. This looks like maybe it's got the simple kind. Yeah. That's not nearly as exciting as I thought it was going to be. It does have a big hook. And then it's got junk on it.
Yes.
It's another pediastrum up here. Piece of one. So those are not diatoms, they're green algae. Again, soft-bodied algae usually don't make it through hydrogen peroxide, um, so it's weird to find them preserved. I know that we put these through hydrogen peroxide. Carlin processed them. Sometimes we'll also use nitric acid, that's just a Olicosyra piece.
Without any central port?
I don't know that there's anything on the inside. I think it's just flipped over. We're looking into the middle of it. Oh, there's a big one.
Yes, it's Nericula.
It is. I think it's Aurora again. Something like Aurora. And this one's Oblonga.
Mm-hmm.
And that's the Stephana discus. It's just cracked open. So, is Anna still out there? Anna, are you still watching? I have a question. Because it's getting close to the time I thought you said you were going to go to Yellowstone with your family. And I'm curious if we're going to get samples of our Idaho Gompho monsters when you go. This one is?
Just a minute.
You want to wait and see?
Yeah.
You have a question about what it could be.
Pinnularya.
It's a Pinnularya, yes. You had it right from the get-go. I don't know that there's anything in there for me to focus on. Alright, I'm going to rotate it. So it needs to go this way, so like
Oh, now I understand.
see? And I just guessed correctly. Or at least I think I did. Don't ask me what species of Pinnularya it is. I have no idea.
Okay, at least you have an idea about this.
There's maybe like a dozen Pinnularya I could identify to species without a book.
I know.
That's and to me, I think that's still an impressive number for Pinnularya, but because they all look similar.
Mm.
Yes, more or less in 2 months. Okay. You're still convinced that I'm going to be making you cookies. What kind of cookies am I making you? If you can collect some of the Gompho monster for us, which I'm hoping you do, happily make you some cookies. I need to know what kind of cookies, though. Let's see. You'd be watching the show and then just dubbing my voice over the diatom names. That would be excellent. It's a great idea, Keelathon. No, we're not. It's not from Yellowstone. They're on their way to Yellowstone, and they'll be passing by some other place in Idaho, where she can collect stuff. Yeah, ginger kind. Okay, any kind of ginger cookie. You want ginger snaps, or you want something that has ginger in it? Like pieces of ginger. I mean, we can make gingerbread cookies. Those are good. And Carlin makes gingerbread cookies with like real ginger. You prefer snaps. Okay. Well, maybe we'll do gingerbread cookies and then I'll make some into diatom shapes.
Wow.
Because I probably can. I might
So
have to
you
get an
need that-
X-Acto. We'll roll it out and then I'll cut out some shapes with an X-Acto knife.
Okay.
You know.
Then for me, do the coconut shape.
What? I'm only making them for Anna.
Oh no.
They're not a little small. I'll just make them diatom sized. You know,
Dart-shaded.
I'll use my stereo microscope and I'll just carve them out with little needles. And then I'll be like, here's a dozen cookies. Here's a dozen cookies for you. Dust-sized gingerbread cookies. No, they'll just be in the shapes of diatoms. They'll be full size. I don't... if it gets me Gonfla Monster diatoms and enough of them...
In that cookies, so do you mix egg too?
Oh, that's a good question. That's a Carlin question. I don't actually remember the recipe for gingerbread off the top of my head. I'm not friends with the gingerbread, so not the recipe anyway. The cookies I am.
Whenever I go to Walmart and I feel like to eat something, I need to ask, or I need to see the ingredients whether there is any egg or not.
Yeah.
Which is kind of very irritating for me. And most of the item it has egg, and okay, it's not for me.
It limits your cookie intake, is what you're saying.
Everything, cookie, muffin, cakes,
Uh,
Chinese foods.
I don't know what to tell you about that. All right. Now I've got one for you. This is a puzzler. Do you know it?
No.
I feel like you do. You just didn't want to say it.
And to see the whole picture.
Here's the whole picture.
I knew.
70.
I knew it.
You know it?
Yeah. Because that middle portion.
Yeah, the middle portion.
But the name.
Well, that's what I'm asking.
I understand. That's why I was silent. Irritating now. It's not coming in my head now.
You said it earlier today. That's why I actually asked. You said this word. The word for the name of this one.
Wait.
And it's the structure.
No.
It does start with an S. You had it.
S?
Starts with an S.
Starts with an A?
Yeah.
No. No. I only told about Storus iron.
I don't think so.
Yes.
I think you guessed something with an S. This structure, if you knew the name of the structure, then you'd know the name of the genus.
Yeah, but right now it's not coming in my memory. What's the name?
This is a Staros.
No, I gave it.
Oh. This is a Staros, and this genus is Staranese.
Storonese.
Hmm?
You told that name.
Did I?
Yeah.
Oh. No, I think I said Staros Sarella. In any case, this is a Staranese.
Storonese. Okay.
Yeah.
this sample, is there any epithemoid species of the...
Epithemia?
Yeah, epithemia.
I think so. We can go looking for one. You want to see them?
Yeah.
We'll take a look. I don't know. I'm not seeing any.
No.
But that's in her previous photos, so we'll take a look.
Okay.
We'll be on the lookout for one.
What's that? That purple one. Let's see.
What purple one?
Here it was showing.
Oh, in the corner?
Yeah,
I missed it.
it's gone.
Let's see. This
Yeah,
one?
Yeah.
Tintinid. It's from the marine realm.
Oh.
It's not a diatom.
This one you posted, I remember.
This one, yeah, that's a diatom.
Some kind of Greek letters. I forgot. This one I like.
This one's Discostella astericostata, and that one is this, that's the Cyclostephanos we saw from Tanganyika.
It's Chorocyra?
Punctus striata.
Oh, no.
Yeah, that's a tricky one. That one Addie's in Addie has in her samples.
This.
No, the
The
Punctus
punctus
striata. Yeah, not that species, but maybe that species too. But they're here in these samples.
Addie mostly bought Chorocyra.
And Punctus striata.
Yeah.
Yeah.
Most of the time I saw her.
She has a lot of Oligocera too. The sample's probably half Oligocera or something like that. But this one is Staranis in turn. So probably at 3 when the stream's over, I'm going to go see how my daughter is doing with the swim lessons.
Yeah.
Hopefully Mallory has not drowned my daughter. If so,
She
I
will
will-
never ever come back to the swimming pool. I have hydrophobia.
Oh, you're afraid of water?
Yeah.
You don't like swimming?
No. Like, my mom tried and I cried very badly.
Really?
Yeah.
Was it recently?
No. That time I was in class 5 or 6 something.
Okay.
But whenever I feel like some shark or snake will come and bite my legs. So
You realize they don't have those in swimming pools, right?
I know, but somehow it's like, oh no.
The swimming pools are filled with chlorine, so there's none of those things in them.
It's very awkward. Like, I don't like swimming.
You don't like swimming.
Mainly, I don't like that water.
Because of sharks.
Yeah. But I like scuba diving. That is mostly awkward.
You like scuba diving.
Yeah.
You've gone scuba diving, but you won't go swimming.
Yeah.
What?
Like, completely separate.
Those aren't
completely
separate.
Completely opposite, but yeah, I like scuba diving, but I don't have guts. So I only like pictures.
Oh, you like pictures of people scuba diving. Okay. I was going to say, how are you scuba diving without swimming?
No. I will never ever go to that under the ocean or something.
You don't want to go under the ocean.
No, I don't want to leave my. I don't want to die over there.
Well, I don't think anybody wants that. Scuba diving I've done before.
Oh.
This might sound surprising, but the most important part for scuba diving is just breathing and not panicking. So maybe it's a good thing you don't do it.
Yeah.
Again, some green algae, some soft algae in the sample. And I'm going to image it. Addie never images it. She skips over them every time. And I'm just like, that one looked cool. Nope. She's here for the diatoms. She doesn't want to learn about the green algae. It's having a hard time figuring out the brightness and contrast, though. It's gonna look good when it comes through. You're gonna be like, we should take a picture. It looks nice.
I thought like this one. I thought like lumps of coconut.
It's not a coccolith.
Yeah. Like later I understood.
That's again pediastrum. You see, each one of these has two little pieces pointing up off of them. Each little cell. That's a good looking algae, given the fact that we didn't process it for algae. Alright, I'm going to collect that image. That one will probably go on to Instagram later. And
With
then
some color? Like
Addie will know that yeah, I'm going to add color. And then she'll know that I had her samples on.
I love the color schemes. I will tell to Addie today.
This is pediastrum that we're looking at. It's not a diatom. It's green algae.
Yeah, but I will tell to Addie that.
Oh, you're going to tell her
Yeah.
that we looked at her stuff?
Yeah.
She won't care.
She is very cool.
She's cool all the time.
Yeah.
Thank you for the follow, RWX Rob. We appreciate that. Let's see. What is the scale for processing sample? I'm not sure I understand the question. Normally when we process samples like these, they go into hydrogen peroxide at 30%, and they sit for a couple of weeks digesting, which is, we know why this sort of stuff usually doesn't make it through.
I think she's asking about the scale of that. But it's changed, like, right? The scale for each species?
The scale of the sample that we're looking at,
Mm-hmm.
there's a scale bar on it, so we can see it down here.
10 microns.
That's 10 microns. The whole field of view is about 50 microns. Little Chook doesn't swim, she just floats. I mean, she's also a chicken, though. Like a literal chicken. Devil and Mrs. J says her son has been taking swimming lessons for 4 years, and it took him 3 years just to build up the confidence to jump into the pool. Devil and Mrs. J, Sylvia wanted to take swim lessons like 2 years ago, but she wouldn't put her head under the water. So, like, everything had to be like this, like, so she wouldn't go under the water. She didn't want her face to get wet. And then subsequently, she's managed to figure out that she can get her head wet and her face wet. Yeah, so. Okay. Yeah, it's a pediastrum. So pediastrum are identified by having two little horn-shaped projectiles sort of poking out of each of the cells in the colony, and then they basically have like a kaleidoscope sort of a pattern. They're a little bit more obvious in the light microscope, yeah. Anna says she likes scuba diving more than swimming too, and actually she only likes to scuba dive. Hey Pacific. I like scuba diving except for the last time I tried to scuba dive, I was in Lake Malawi,
Oh,
and it was where the hippos were. And I mean, there weren't any hippos there, but it's where the people had previously seen hippos, and it was also like where the mouth of a river came into the lake, and as a result, it was murky. And I was like, "You want me to get in the water with the hippos, and I can't see what's going on?"
no.
Nope. I mean, I got in, and then I was like, "Yeah, I don't feel comfortable with this." The hippos, you don't want to mess with them. I'm just saying, like, not that I think you want to mess with the other choices, which are like, crocodiles or.
I don't want to mess with any that water, like that kind of animals.
Well, I have news for you.
But
None of the animals were sharks.
but crocodile is nothing, not less than dangerous than so.
I don't know. Like on a scale of which one kills more people every year, is it crocodiles or sharks? Probably crocodiles.
Both are harmful. So I want.
Both are harmful, yeah.
So I want to avoid both of all harmful species.
But people die from crocodiles on land. So.
That is their over smartness.
Well, it does have something to do with how.
Like they
Where they put themselves.
Yeah, they sometimes cannot identify while in the beach they walk or they take rest, and crocodiles may be beside of you.
Yeah, I don't think the crocodiles bother people when they're sleeping. I think you have to kind of mess with a crocodile to make it.
I don't believe them.
You kind of have to like be
They're
where
very
you shouldn't
cunning.
be where you shouldn't be for a crocodile to like do something to you. They're cunning.
Yeah, they are very cunning.
Crocodiles?
Yeah.
I don't know if I believe that.
They look like very innocent, but they are cunning.
They look innocent.
Sometimes.
I don't know what that crocodiles look very innocent to me. They look like logs with teeth. I don't think they're hiding that from anyone.
I hate those species, those animals.
The little ones, like the caiman and the, I don't know, I guess baby crocodiles, they're cute. You don't think they're cute?
No.
Oh, they chirp. They're like, nah, nah, nah. They're cute.
I don't like tiger, lion.
Tigers?
I don't like them.
You don't like anything with teeth that might bite you.
Yeah.
That's what I'm getting from this.
Harmful there.
Well, I mean, there's a bunch of harmful little things that you can't see. What about those?
Is there anything? No. I
Not-
thought like something here.
In the room with us? I don't think so. She's like, you're talking about crocodiles, and now I feel crocodiles are in here with us.
No, you told me that little things also can be harmful.
They
so-
can. Yeah. I mean, I gave a whole lecture on parasites. You can hardly see most of those.
I saw one lecture, I think.
Well, it wasn't about the parasites, though. Those ones, they could potentially harm you pretty badly.
Though cockroach and spider are not harmful, but I hate them too.
Man, it's starting to be a long list.
Because they always live in a dirty place.
They live in a dirty place.
Yeah.
Well, they can't help that. Spiders don't necessarily live in dirty places.
They looks like very ghost type.
Spiders can be harmful, though.
Yeah.
Usually
That's
only if
true.
you mess with them. There's not a lot of spiders that creep up on people and attack them in their sleep on the beach.
I never mess with anyone.
You never mess with anyone?
No. I'm always kind of peaceful person.
Huh. Well, Probably you don't have anything to fear from spiders then. I found another
I
pediastrum.
only like birds, dogs, cats. I don't like.
You don't like cats?
No, sometimes they behave like very selfish.
They're selfish.
Sometimes,
Cats are selfish.
yeah.
In what way?
Because one day I was eating my fish and that cat came and snatched from me.
It took your fish?
Yeah,
And now you hate all cats?
yeah,
Because one cat took your fish.
yeah.
Maybe there was something wrong with the fish and the cat was trying to help you.
No, cat loves fish.
I know, maybe it was harmful for humans though. And it was trying to save you from it.
That fish was not harmful for us.
You never
It's
know.
river fish.
You never know.
It was my favorite.
Oh. Well, I guess maybe you do know then.
Yeah. I like only birds. And dogs too.
Yeah, Anna saw a moose the other day. Walked right through her neighborhood. What about mooses? How do you feel about them? See? Look at this. Little Chook says baby croc noises are so cute.
Ah,
She knows. She has a false black widow living in
don't
her
call
garage.
their trap. That is all their trap.
The cute noises are part of their plan.
Yeah, to attract you and then kill you.
See? On the diagonal says science suggests that both baby crocs and baby birds make cute noises. Maybe baby dinos did too.
Oh my gosh. Okay, but I don't want to try crocs and dinos.
You're afraid of dinosaurs?
Everyone, I think.
No one's afraid of dinosaurs. They're extinct.
Now they were extinct.
I mean, they've been replaced by birds. Are you afraid of birds?
No,
Birds are just dinosaurs.
but both of them are separate, like different characteristics they have.
I don't think that's true.
Like birds never kill you.
I don't think that's true either.
Birds can kill us.
There are birds that can kill people.
Which bird?
What?
I want to avoid them then.
Oh, like ostriches could kill people
Oh,
and emus.
so that means I understand like big sizes harmful. Like big animals.
Why'd I say that? Now she hates birds. There's fish that can kill people. How do you feel about fish?
I like only river fish.
Only river fish.
Yeah.
So I'm going to do some research on which river fish can kill people.
Okay. And here I'm not getting enough river fish.
You're not getting much river fish here?
No.
It's probably a good thing because the rivers here are highly polluted.
Yes.
Probably best if you don't eat from them.
But I saw like many tilapia fillets here. I don't know. Is it river one?
No, I don't think tilapia is a river fish.
Then it's good. Because most of them I eat. Most of the time I eat them.
I don't eat fish at all, so
Oh.
I'm probably the wrong person to ask.
I love fish.
I feel like I should just scroll around and just take pictures of the green algae
Mm.
because Addie won't do it. It's all the same big ones.
Yeah.
All the big ones are the same.
But diatoms are not harmful.
There are harmful diatoms.
Oh.
Just one genus that we know of,
Oh.
which is Pseudonytzia. They're marine. So you don't have to worry about it
Oh.
because you don't go in the water anyway.
Yes.
But they make a toxin that you could potentially consume. That one looks kind of trashy.
That's close. It's
That one was a navicula. It just looks kind of trashy to me. It was covered with clay. This is also
and very green
pediastrum.
energy.
Yeah. I'm sort of curious what pediastrum comes out like in the
Oh,
closed captioning. Let's see what we can do about finally fixing my image now that we're almost done. To the point where we're almost done streaming and I can finally maybe fix the quality of the images a little. No, it's not actually that bad.
it's good.
See? These should look like little salt and pepper shaker heads, and they do. I always feel like they could do better though.
Which one? Like the image quality?
Yeah. I mean, find one that's kind of, yeah, like in here. We'll lower the... I think my stigmation was actually pretty good. We'll just put it somewhere in here so they're all on the outside edge. So we get a little bit of all of the valve face photoportula. They look like they're in good focus.
Yes. So next streaming is on Wednesday.
Yeah, our next stream is Wednesday from the SEM. And I think it's going to be, I have guests that should be here, Mark Edlund and Joe Mohan. They were supposed to come, I thought they were going to come this Wednesday, but they actually are going to, I think, be here next Wednesday. So, hello, Adrian. How's it going? Let's see. Ostriches are crazy. Yeah. A piranha are fish that can kill people. Yeah, that's a good. They're little. They have big teeth, but
Is
they're
it
little.
Like river fish?
And electric eels are freshwater fishes. Oh, they say piranha mostly eat fruit.
Oh, it's river fish. Oh,
Yeah,
no.
piranhas are river fish. Yeah. And they've got big teeth, and they bite people.
Oh my god.
Take your toes off.
Oh.
Diatoms aren't big enough to hate. Salmon are ocean fish, yeah.
Yeah, salmon, good.
Yeah. Pediatric. How are you, Adrian? It seems like maybe your caps lock is stuck on. Is that the case? Anna just added a new diatom to her life list. How many diatoms are on your life list, Anna? I don't even want to know how many are on mine. I mean, I probably could just name like 400 diatoms that I know off the top of my head that I could identify. So, like, That's way more than I can do for birds. You don't count them, but if you made a list, the list should be able to count, right? So it's a conceptual list, not an actual list. You do count birds. How many birds have you got on your life list? What are your bird totals at, Anna? The diatom list is just in your head, okay. I mean, every time I start a project, I find 528. Dang. That's up there. You've got like 300 more birds than me. Although, to be fair, you know, you've been on multiple continents, so... I mean, I haven't really been able to go birding when I was in Africa, or I probably would have a lot more.
What were, oh, okay.
Yeah, this is Niagara.
Okay, someone asked, what are the white spots?
I guess it depends on which white spots they mean.
I think this one.
These things?
Yeah.
These are valve-faced photoportula, sometimes called strutted processes. And they're a special process on the diatom. There's a hole that goes through to the outside world. I don't actually know what, like sometimes, for some organisms, the mantle photoportula have a purpose where they create sort of a baffle as they sink by putting some EPS out them, or no, chitinous material out them. But the valve face ones are a little bit of a mystery to me. In some diatoms, they use them to connect in colonies to other diatoms. But I don't know that Stephanodiscus lives a colonial lifestyle at all. So I don't really know why Stephanodiscus needs them. I'm not positive on the answer to that. What do they do sort of question. It's still a bit of a mystery to me.
This one from the same Clear Lake?
Yeah, we're just going a little bit farther down in the core.
Okay.
So these are all from Addie's stuff in Clear Lake. I was trying to see if I could find an epithemia for you.
Here one in cyanide.
Yeah, I think that's Cimbella.
Huh?
I think it's a little too big for an encynema.
Oh, sorry.
But it's just an outline, so it's hard to tell. So nobody's right until we actually see a valve. We can zoom out and see if we can try to find one, though. I think it's this one that we looked at. But the valve ends are missing, so... If I had to guess, though, I would guess Cimbella, not encynema.
I think you are correct.
Well, if I'm not, nobody can tell me otherwise, I suppose, except for Anna.
Who is she?
Who's Anna? It's Anna Nastrovich. She's another diatomist.
Okay.
We're trying to see if we could get Anna to come work here, to be a postdoc in my lab.
Wow.
But it requires grants to be funded, so... She wants to look at all those nasty Nitsia in Tanganyika.
Then I want to join with her.
The two of you can have it. I don't want it.
Because I really like Nishia, but nowadays, from today, I have doubt on my...
You have doubts about whether you like them?
Yeah.
Just starting today?
Yeah, but I will again come some another day and try to find it out. And if I can properly, then I'll like again.
Oh, I see. You only like things if you can identify them.
Like, they're making me confused. So, why do I like them?
I don't know. I think they're easy to identify that it's a Nitzia. Like, to get it into the genus is pretty easy.
Yeah, but the species level.
Yeah, species level is a challenge.
It's microscope.
I think it's a microsphere. There's also chrysophytes in the sample, though, so
Oh.
but I think that's a microsphere.
Mm.
Hard to say, because sometimes the aperture is pointed away from us. It's like a big piece of a leaf right here. Yeah. So when you see those like long crenulate phytolith cells, they come out of this part in here.
Mm.
And when you see those sort of saddle-shaped ones,
Mm-hmm.
they come out of this part right here. See them?
Yeah.
So they're in their life position, which is kind of cool. I always get excited when I see these kinds of shots where you can see the cells and the structure of the phytoliths in them. And I get a picture and I take it and I show it to Chad and he's like, yeah, no big deal. He's never excited about them. I don't know why. I guess he doesn't I guess he doesn't care that I took a nice picture of a phytolith, so as a result, I just stopped taking pictures of phytoliths. Chad's not excited, I'm not excited.
Yesterday I was looking some fecal spherulites, and
You were looking at fecal spherulites,
yeah,
and
and while looking at them, I continuously asked actually, I was like, from which animal this poop? And every time I'm scared, if it's human, then I will not want to see.
you don't want to see it if it came out of a human?
Yeah.
Why not?
Yuck.
You don't like that?
No.
Well, most of them probably came from humans in his sample sets.
But I think it was not from human.
Why do you say that?
He told me.
Maybe he just said that, though. Maybe it's like the fish.
Ah, fish is okay, but I don't want to see humans poop.
No, I mean, maybe it's like he knows that if he tells you it's a human poop, you won't want to look at it.
Oh no.
So maybe
I
he just
don't
told you
feel-
that.
No, he didn't understand. Like, I don't like human poop.
You don't like human poop?
No.
Well, that is shocking. Well, I think on that note, we'll probably end for today. I'm going to zoom in, maybe take one more picture of Aulacosira here,
Mm-hmm.
and then I'm going to go see if Mallory has drowned my daughter.
I told her, Mallory, yesterday that I'm gonna see you how you train Sylvia.
Well, you should get lessons from Mallory at the same time. That would be great. And
Yeah.
Sylvia would have a swim partner.
Yeah, that's the good idea.
You think so?
Yeah.
When,
I need to now ask Mallory what's her schedule so that I come.
that's a good plan. And then you could just hop in with Sylvia
Yes.
and put you some of those water wings on. They'd look good on you.
Yeah. I'll also come with you today to see Mallory and Sylvia, especially Sylvia.
Okay.
I never met him.
Oh, yeah, that'd be
Met her.
good.
But she is very shy girl. Like me.
I don't know if that's true.
I think I was new to her, that's why she was feeling shy.
Yeah, she's shy sometimes, but, you know, also she can be a handful. So, these look like they're doing okay. we'll get a 2 cells. I don't need the whole chain really. And then slow it down. We'll do this. See if anything exciting's happened here. Let's see.
You have to challenge.
That's what an extra continent can do for you. Yeah, that's true. How do colonial diatoms connect? Do they form hard or soft bridges or just vacuum onto each other? Most of the time it's hard connections like these ones. So the one that we're looking at in the image right now that you're seeing is an Oligosyra. And then these little sort of triangle-shaped things that are interlocked are linking spines on the Oligosyra's valve. So their skeleton actually has hard parts that are interconnected. But that's not actually necessarily always the case. Some of them have sort of specialized pore fields. So they put some material that basically allows them to connect. And then some of them just have sort of like a thread of biological material as well. So it's not consistent. Yeah, see, she wants to look at all the Nitsia. Jonathan Gamecast said, I left a child with Mallory. Well, this is not totally true. My wife and my child are with Mallory. So Carlin is there to supervise and hopefully keep Mallory from drowning my daughter. Yeah, it'll be okay. All right, so it's 3 o'clock, and it would be a good time for me to find somebody to raid so we can end the stream. I want to thank everybody for hanging out and, chilling with us. Oh, Ursa Crafter's on, and that's great, because I haven't had a chance to actually raid Ursa. Oops, I need to turn that off. She's an artist that was drawing some stuff that her and John Renzella were drawing during a co-stream last week, and so she does drawing and other art on Twitch. So we'll give her a check out, and hopefully I got that right. Yeah. So we'll rate her, and I want to thank all the people who followed today as well. So it's Holtzy and None More Grim and Rob. Thank you guys for the follows, and especially want to thank, as always, Pacific Plankton for hanging out, and Anna for showing up to chat with us, and Devil and Mrs. J and Little Chook for hopping in, and Jonathan's Gamecast. We didn't give you a shout out, but we should. Hang on. Should also check out Jonathan Gamecast. There you go. And everybody else for just sort of hanging out and chatting with us. Kalathon and Bluesy. It's nice to see you guys hanging out with us. And Dangling. A lot of our old friends are here in the chat with us today. And yeah, as Pacific Plankton has sent a link for, you should check out some of these other streamers. Spider ID's on right now, but he's playing D&D, so that's why we didn't pick him. So, all right, we'll see you guys soon. Wednesday we'll be streaming again, and now we're gonna raid. So we'll catch you later.